everyone for coming back. Let's move on to our panel discussion. So I think we had like 5 really fantastic talks to start off this session, and I guess everyone, I guess all of you are ready with many burning questions in mind. So I'm gonna go ahead and turn it into someone who will start with our first questions to our panelists. Or if not, Jim, do you want?
Yeah. Okay.
Oh, yeah. So
yeah.
Please.
Thank you for all the wonderful talks. So I have a general question that I think pertain to all the talks, and that's the problem that we are becoming aware of, heterogeneity of tumors. And so some of you address that. So for example, the neoantigen is probably expressed only in a minority of cells. And how do we deal with that? And do we just rely, there are these theories of innocent bystanders and epitope spreading and so I think that has been pushed aside a little bit, and can we confront that problem in a more direct way?
I think that's a very important topic, and I've been working on cancer for 30 years, and I think that's probably the number one thing I learned, how heterogeneous tumors are, cancers are, and also I work in pathology department at MD Anderson, and then I have plenty of pathologists reminding me every day how heterogeneous cancer are. Because if you look at under microscope, just one tumor, different part of a tumor, they can look very differently. It's just tremendous. And actually, when Bernard talked about the fluorescent staining to look at the spatial relationship between cells and immune cells and tumor cells, and I just say, wow, this is very, very important. The pathology will love you. I tell you, they think this is the most important thing to do, but it's also very challenging, because if you look at the different part of the tumor, the spatial relationship can change, and I don't really know how to deal with the issue. And because for all the study we do, we always sample part of the tumor. So we never get a complete picture of any tumor. Any tumor we look at, pretty much we only know the partial information. And that's also explained why the pharma always complained in research, and like 75% of research publications are not reproducible. I think this is because we're dealing with heterogeneity. You're not going to get 100% reproducibility. It's just not going to happen, okay? Because every report is part of the big picture. And if anyone looking for complete reproducibility of research data, you're dreaming. It's not going to happen. Yeah.
I'm talking about the intratumoral heterogeneity, because that should be reproducibly bad, right? It should not work if we have intratumoral heterogeneity. But it still works. I mean, immunotherapy works, precision medicine works. So I think it's more important for us to not just push it aside and be thankful that things work, but ask why does it work if in theory it shouldn't work, because individual cells are different. And I think we might be able to learn something from that.
Yeah. Well, I think we're all looking at what the heterogeneity is. Our plan is to target antigens that are unlikely to be heterogeneous because they're critical for maintaining the malignant phenotype. But we've already seen with that there still are workarounds, and so potentially targeting one oncogenic protein may be inadequate. But I'm going to give this to Alex and Bernie, because basically they are dealing with the heterogeneity by providing a polyclonal response that hopefully does capture that heterogeneity, so that you may not express one antigen, but if you have a polyclonal response, you theoretically can.
So another part of this, I think probably all of you know about the theory of immune surveillance, right? The reason that we don't get cancer is because our immune system is constantly surveilling for things that develop. So this paper from Tripathy, which was PNAS 2016, MD Anderson, Ignacio Wistuba, Jeff Muldrum, a bunch of other people were on it. It's really an amazing paper. It's just mistitled. That paper shows, to my way of thinking and my interpretation of that data, is that their patients have got a large number of antibody responses to epitopes, to proteins whose peptides could be eluted from class 1 molecule, and they could show CD8 T cell responses to peptide after a brief in vitro sensitization that was like 50% killing at 20 to 1 E to T, and that's a lot of detail, but I mean, it gives me some strength, some encouragement that this is actually real, and we've actually repeated it in mouse models and have seen the data that was published last year out of my lab. Tyler Hewlett was the first author, and we're continuing to push along with that on the patients that were enrolled in our study for non-small cell lung cancer, and we're trying to get there. So we've got a collaboration now with Shimadzu, where we're actually looking at the peptidome in both breast cancer, and actually worked with Paul Spellman up at OHSU last year to look at a whole series of breast cancer cell lines to identify the peptidome, which are the antigens and epitopes we're going after in terms of our breast cancer vaccine study, to see if there are pre-existing antibody responses to these proteins, so we can then maybe boost or look for augmentation of these things. So I think that heterogeneity is real. I think, as Phil mentioned, I mean, if you look for driver mutations and epitopes there, you're going to be able to go after everything potentially, or a large percentage in some number of patients. But I think from what I really think is that if that's true, and I've been talking this up a lot to different groups, and I think it's very provocative, but it's stuff that we really already know. Immune surveillance is an important component. It's failing those patients, but the fact you can separate their T cells from their milieu in vitro, stimulate them briefly, and get killing tells you there must be ways to get around it. So maybe it's the lipid accumulation that you showed, right? Maybe there's other things that are happening that are turning them off. But I think there's things we could do. And so I think the other strategies you heard today touch on different ones. I mean, I also like the iNKT, because both NKTs, NKs, they give you other whole realm to go after cells that lose class 1 or lose antigen if you've got enough of the response.
Okay.
Can I follow? Sorry, Alex, were you going to?
Yeah, I was going to add something. So I turn your attention to the body of work from the Cancer UK, Charlie Swanton's work with over 800 patients evaluating inter and intratumoral heterogeneity, and the papers that have been published in a variety of different high-profile journals that looked at the phylogeny of mutations both in the primary tumor, locally advanced tumors, and metastases from the same individual. And I think the data that's emerging is very clear at showing that, and Charlie Swanton has made a lot of work on this point, is that there are truncal mutations that are common to all tumor cells. Now, we can understand that if from germline, tumor is evolving, and there's a whole collection of tumors that participate as part of the trunk of the tumor, and that later you have evolution of passenger mutations that are going to be individually separate. We can evaluate from analysis of, in spatial as well as longitudinal studies, whether tumors harbor these truncal mutations, and the evidence to date suggests that the answer is yes, and that it's possible to witness in a multiple of different tumor types that those mutations that are common to the tumor remain common to the tumor regardless of the evolution of that tumor.
I wanted to follow up on the intratumoral heterogeneity question. So how about non-genetic intratumoral heterogeneity? We know that also exists. And so what are some of the strategies that all of you can envision in dealing with that in terms of immunotherapy approaches?
Well, so there's actually been some beautiful work that's come out of Aviv Regev's lab recently. Now that you can do multiplex analysis, as Bernie talked about, in tissue, and indeed the limits are not clear yet, but you can do, for example, look at least 50 different genes that are expressed. What is clear is that when you look at tumors that many of which have initial responses to immunotherapy, there are actually geographies that are going to be resistant. So there's this common description now of tumors that are called hot or cold. Hot being that they're inflamed, there's T cells in them, and cold being that they're not. And what her data is showing is actually, if you look at the whole tumor biopsies, there's actually geographic regions that are that. It's not actually that the whole tumor's hot or the whole tumor's cold, but that there really are regions that are cold, and that would predict that, for example, with strategies such as checkpoint blockade, you're going to have areas of resistance. So the issue, of course, is now to understand what is that heterogeneity about. There's data, right, that it's the beta-catenin pathway is an important way of excluding T cells from entering. There's also data that CDK4/6 are overexpressed, and indeed in most of the islands that she sees that exclude T cells, they have that. So fortunately, we have drugs now that can target some of those pathways. So as we understand more about what that heterogeneity is and how that impacts the efficacy of therapy, we'll be able to combine immunologic manipulation with pharmacologic manipulation and try to achieve more global responses.
And go ahead.
Yeah, I would like to add one comment. So the intratumoral heterogeneity, the epigenetic changes, there are many different forms. There can be like a chromatin remodeling, there can be methylation change, there can be alternative RNA splicing. So all those, that's what the immune system is designed for, to monitor all those changes. So all of those changes eventually will reflect either protein change, right? Either sequence, new epitope, all of those, and lipid change, right? So that we just talk about this. Part of the immune system are designed to monitor, like each part of those. And because of the way the heterogeneity of cancer, and because of the way the fast mutation rate were happening, all along, it's not stopping at one point, it keeps evolving like daily. So the unlikely a single therapy, even immunotherapy, a single therapy will be, one cure, for everybody. So people are talking about a combination therapy, right? Like the cocktail therapy for HIV, that's eventually whatever worked to control the disease. And likely, for the cancer therapy, it will be combination. It can be radiotherapy drug, chemotherapy drug, or kind of small molecule drug in combination with the current form of the immunotherapy drug. And hopefully all of those, the cocktail therapy can really control tumor.
And I was just going to mention, following up on Phil's comment and your comment, Lili, it was the digital spatial profiling, and I'll get it probably wrong, but I think Joe Beecham at the AMP meeting was saying they were doing up to 1,600 transcripts, right, in that single cell or 300 micron area. And so I've been really fascinated because you do see these differences, right? You see areas, the intratumoral heterogeneity, where you don't have T cells going in, and you've got macrophages that don't have PD-L1, but the macrophages there, what are they making that's excluding it, or what are the tumor cells making that are excluding it in those regions? And so I think we have a collaboration, and Sarah Church, who's in the back, told me that she's got the data, she did the IL-360 panel on it as well, and so we're going to be able to, and we put in a bunch of other genes that we were interested in. But I think that's the kind of things we're going to be able to do, and I think if it's not going to be easy, I'm sure, but you take something like mesothelioma, nothing seems to work, everybody dies in that disease. What's going on? Why can't, mesothelin, great antigen, why can't you just do that? Why can't you get something to work there? So I think there's got to be something else that we don't get, and I'm really optimistic that by doing these kinds of studies, we'll get other data that'll help us figure out strategies.
Yeah, Lili, I think so TCGA, we have been very mutation centric, and we have not paid enough attention to alternative splicing, for example. You may not actually see mutation, but actually you generate different protein forms, and we don't really have very good technology to look at post-translational modification, glycosylation, for example. And I think all this going to, not being an immunologist, but I think all those post-translational modification can impact the immune response. And actually, I want to hear from the panel, like, what do you think of those impact of post-translational modification to the immunity?
Well, clearly it can modify, but fortunately now the technology's marched on, right? So now you can do site-seq, right? Which is essentially you put an oligonucleotide on the end of an antibody, so you can actually look at protein as well as RNA, so that we're not stuck now only looking at message and trying to infer from that. We can actually look at the heterogeneity of proteins, and as long as you have an antibody, if you know that you have a protein that when it gets modified changes the biology, you can actually look for whether it's present or not present. So I think the density of information that we're going to be getting is going to be increasing logarithmically, and a lot of this is going to require actually high-dimensional data profiling, and that's going to really require a lot of interaction with bioinformaticians who actually can tell us how do we interpret now all this data that we can collect.
Okay. So let's move on to our next question.
So with all the heterogeneity, how do you ever do a clinical trial and balance your trial? And then I guess a follow-up question is, with your precision medicine, now that you know different precise parameters, how are you sure you don't have bias, whether you're a precision company or a pharmaceutical company, to know that you're really doing the best you can for all these patients?
I guess that question maybe for me, but and I'm not an oncologist. Let me first make a statement. However, I think the clinical trial, there are a lot of discussions right now, and I think FDA actually they've been talking about how to change, how the clinical trial is set up, and exactly because of what you said. And tumor is very heterogeneous. And then whether a clinical trial is going to be successful or not, it's a game. It's all dependent on what patient you're going to select for the trial. And then so you can generate beautiful result and p-value is significant and then the published New England Journal Medicine paper. However, after you get approval, when you go to the real world, the real patient community, and then the patient you treat, they don't really match the patient in many of the clinical trials. That's why I think we're going through the up and down emotion all the time. We're excited about some clinical trial being successful, and then when you go to the community in the real world and treat all sort of patients, and then you have all those compounding factors because of the heterogeneity. And then we have not had a good solution to solve that bias. I don't really know the answer, and I think a lot of oncologists have been discussing about this.
So if I can just add to that, in the FDA objectives, which were July 20th, it's the FDA CR. It was the dose finding workshop, but there's all the data. You can find it under the Center for Oncology at FDA. They're really pushing right in terms of being able to stratify patients more efficiently. So how are you going to do that? So being an immunologist and not a genetics person, we're going to do it by the kinds of things I was showing you. It's going to be looking and evaluating T cells and where they are. So how are we pushing that forward? So I presented to you the data that we published in Lancet in May, which was the result of 6 years of work of cajoling pathologists who I love pathologists. I spent a long time in surgery departments, but I love pathologists because they know exactly the way to do it, and even when you tell them the way they got to do it, they do it their way that they know works, right? So you got to kind of get them, and it's the same whether they're in China or Japan or Korea or North America or anywhere else, right? But once they do get on board and you get the results and they're reasonable, you got a way to change the classification. And so I think what we did in validating that data for going to the Commission on Cancer and trying to push forward with AJCC to change the way colon cancer is staged. I think the data is there. I think we can do it. There's more data that's going to come out. But that's going to start with an immunological component to staging in colon cancer, which I think is real. So the data in a science paper in 2006 was published, showed that stage 2 colon cancer, for which you don't do anything typically, if you've got a very low immunoscore in those patients all progressed like in 24 months. So with that data, the Walter Reed, which covers all the colon cancer for the military, the NCI, was interested in integrating, and they've got central review of all the colon adenocarcinomas, so they were looking at integrating that into a trial so they could actually identify patients up front with early stage colon cancer that had a low immunoscore, and we could put them on a trial, an immunotherapy trial, thinking that the reason they're failing is they don't have T cells, they haven't recognized it. So this was involving Bavarian Nordic, some other people, the NCI, Jeff Shalom, and that's kind of halted because the staging hasn't been pushed forward. But I think it's going to be those kinds of things that are going to be are things that the diehard immunologists have believed the immune system is important, but it's getting enough data to convince our pathology and medical colleagues that it's real and we should be integrating this into patient stratification for trials.
I do want to add one thing. When we propose the TNMB system because the pathologists, they look at the TNM, the first staging, and then but they have been complaining about TNM for a long time, so they know they're not good. And when we did the blood liquid biopsy data, at first I thought, well, the pathologist is not going to be happy with our proposal. You have to add another data to make staging. But in contrast, actually, when I talk to pathologists, they all love it. They really buy into liquid biopsy, and so I think liquid biopsy is just is so easy to do. And I think there's just such a rich information you can get from blood. And it may not be from one drop of blood, but from 10 cc or 1 cc, you can get a lot of information. I think those information I really think going to make a major impact on precision oncology. And just impact on how we get the patient information before you design anything. Because you have to stage the patient correctly. Otherwise, nothing you do is going to be clean. Yeah.
Yeah, I think the question began with this: with so much heterogeneity, how do you design a trial, and how do you decide who to treat and not treat? And the reality is, with just a couple of exceptions, such as what Bernie was telling you about in terms of knowing we should intervene in this subset of patients with colon cancer, even though they look like they're early stage and ought to do well, because we know they won't. We don't really know what most of this heterogeneity means. And we don't know how big an obstacle it is. There's tumor cell intrinsic heterogeneity, and there's microenvironment heterogeneity, there's interpatient, just genetics heterogeneity. But that's precisely the reason why we have to be collecting high-dimensional data for in all these trials, because then we can learn when we do an intervention whether or not it works or doesn't work, and we can also try to understand when it doesn't work, why didn't it work? Or when it does work, what was present that made it work? And that will allow us to design better and better trials. But this field is really in its infancy, and the ability to look at geography of tumors now and actually understand that kind of heterogeneity, that wasn't feasible a year and a half ago. So, it's moving very quickly.
Vincent?
Hi there, thanks for all the great presentations, Weston Thorson, ISP. I'd like to go back to the question of the combination therapies of immunotherapy and other combinations. Obviously, there are a lot of different possibilities and a lot of different choices. What do you all think would be the best way to narrow down those choices? What data do we need, and what methods would we need to advance that beyond the combinatorial explosion that it is?
I think if we had the answer to that, we wouldn't be sitting up here. We'd be all celebrating and the end of cancer. I think part of what has been discussed up here is high-dimensional data. One of the things that I'm struck with when we do DNA, RNA sequencing is that we learn the profile for each individual about what's going on in their tumor. I think some of the data that you've seen here from some of the other speakers shows that we're learning more and more about what are other implications of the starting point for each individual. And I think rational evaluation of that, perhaps through analysis of big data, and Bernie brought up the fact that we should give credit to to the agencies for being the source of accumulating these data, but then it's up to all of us, all of you, to try to be able to make sense of these data. But I want to start out with something else as a premise. 4 years ago, 5 years ago, we couldn't be sitting up here on this stage talking about immunotherapy because the doubters exceeded the believers. And the fact is that we've now witnessed that, in fact, a single monotherapy with immune checkpoint resulted in profound outcomes for patients. And we shouldn't lose sight of the fact that the doubt that existed prior to confirmation of that data paralyzed the whole field of immunotherapy. And yet here we are speaking about what can we do to leverage all of this information to be able to do better and better. And I think we're getting closer to the point where we're beginning to interpret those data from being able to evaluate high-dimensional data. And it is going to be from sort of personalized evaluation and personalized decision-making between the doctor and the patient to decide what are going to be the most likely things to combine, and not sort of say, because we have these two drugs in our portfolio, those are the ones that we're going to be choosing. So I think we're at the cusp of another revolution in terms of data and data interpretation. I think that's part of the reason why the ISB has such a potential for profound influence in what we do going forward, is the analysis and the evaluation and the interpretation of those data.
Yeah, I think in terms of trying to understand what you combine when, I think we're probably pretty ignorant now about precisely what should or shouldn't be combined. But all of us are looking for biomarkers of predicting what belongs with what patient, and whether and what combinations belong with what patient. And it's only going to be really the retrospective analysis of outcomes from what's done that will actually be able to understand what should have predicted that was going to succeed or fail. And once we have that, then the trials will become more and more rational and have presumably better and better outcomes in the people who receive those therapies. But again, this is just so early in the field that it's really hard to now provide you with some rational story, except for the very few, very clear cases now where we know what does predict and what doesn't predict.
I would like to follow up on the point, like the depth and breadth of the data will be only enrich that richness of that data and the systemic analysis, improve the technology, more data, more information. And those need to be human data. I guess that all researchers, sitting in this room, like immunologists, probably share the same, feeling as me. So we study a lot of things, most of the things, in the mouse model. We know a lot about the mouse immune system. We know the T cell, we know how to culture them, we know how they behave. And if mice are our patient, we probably can treat all the diseases a mouse can have. But that's not true for human. And I guess that Phil and other people on this panel can tell you that's a fundamental difference between human and mouse immunology. So human immunology, the human T cells, for instance, how they behave, it's different from what mouse cell behave. So all those information, like how those cells, behave like in vivo in a patient, those information can only come from clinical trials. And the accumulation of those information need time and need the support of the advanced technology. And as Phil mentioned, it's just a beginning. We just touch the tip of the iceberg to know what's going on. But now, when more and more clinical trial ongoing and incorporating of all those rational design, say the biomarker, prognosis, and what kind of treatment, and analyze the data. It's not just run the trial and say work or not work. All those information, what happened in the blood, what happened at the tumor site, and what happened longitudinal, manner, right? So like all those information, when accumulated enough, we can see the rule. Then we can work on that and work on the possible better way of prognosis, prediction, and what kind of biomarker to use, and what kind of therapy, precision, medicine or combination therapy give to each person.
So Bastian?
I apologize, I've got to leave.
Oh, okay.
So I
Yeah.
really hate to do this, but I really enjoyed the meeting, so thank you guys very much.
Thanks,
I got to catch a flight to see my wife this evening, or I'm going to be dead.
So Bastian, let me. I think that's really important question, and of course, we talk about all the looking for for markers, and a lot of those things in practice actually very difficult to do, and there are many factors. I talk about the challenges, and you have to get biospecimen, you got to do all those assays, and then there's a physician fatigue and burnout. So in practice, actually very difficult to do. But just look. I always want to go back to TCGA because TCGA is such a huge investment, and then we have published so many papers in TCGA, but I think we're still just scratching the surface, and we can look at using other computational biologists. I think can play with this data for years, and their clonality analysis. You can computationally actually determine how heterogeneous a tumor really is, right? Because we say that words not being quantitative. We don't really have a matrix to determine whether it's 1, 2, 3, 4 in terms of heterogeneity. Not all tumors are super complex. There are some tumors that are relatively simple, like GIST, right? GIST, compared to many other cancers, they're relatively driven by some specific genes. They're actually we consider the less complex tumor, a relatively simple tumor. But we just say that in those hand-waving terms. I think the computational biologists like you and like Lili's group, and we can actually develop. You guys can develop tools, computational tools, to really quantify the degrees of heterogeneity. And then perhaps in the future clinical trial, we can start with something simple, right? Something relatively simpler tumor to do the clinical trial. Because only simpler tumor you can talk about combination therapy. If you have like 20 clones, there's no way you can do 20 drug treatment for unless you believe in Chinese medicine, right? It's a mixture of everything, but of course you don't really know. So if you do like evidence-based treatment, combination therapy, I think you have to start with relatively simple tumors, and then I think this is where the computational biologists can make an important contribution, and to find a way to help biologists, help oncologists to quantify how simple, how complex the tumor really is.
Okay, so with that, I would like to thank our panelists for the wonderful discussion, and thank you all for participating.